introduce this speaker. Many of you read about papers and with gens and gens, but most of you heard really talk about channels. And rarely do we have a chance to hear Yuh-Nung talk about channels. So it is with special pleasure to have Yuh-Nung to speak. Yuh-Nung comes from UCSF, many of you already know. So,
thank you very much, and we would also like to thank the organizers for inviting me to this nice meeting. Lydia and I have been collaborating for almost 40 years, and about 20 years ago we became sort of specialized, at least in going to meetings. So she would go to all the channel meetings, and I would go to all the development meetings. Until this year, because our development work sort of led us back to channels, in this case, mechanotransduction channels, and that's what I would like to talk about tonight. So mechanosensation is a very interesting and relatively understudied research area. And among all our senses, the mechanosensation, for instance, our senses of touch, sound, gravity, etc., is the least well understood as compared to other sensory modalities like vision, olfaction, and so on. And one reason is that because of the difficulties in identifying mechanical transducing molecules, so mechanosensor cells, they are not abundant. So for instance, for hearing, there are only about 15,000 or so hair cells in our trachea, as compared to millions of rod and cones for vision. The mechanical transducing molecules, they are of low abundance. So the estimate is that we have 100 channels per hair cell versus say millions or dozens in each photoreceptors. And so the sort of hearing channel, which is the holy grail for people in the hearing field. So in terms of abundance, the channel is like 6 orders of magnitude less abundant than the dopsing. And moreover, assay mechanosensory properties of the candidates is often problematic. And so all this together making it very difficult to identify mechanosensory channels with biochemical or molecular biological technique. So it is similar to cloning shakers. In order to get to potassium channels, perhaps once more we can use applied genetics to try to identify mechanotransduction channel. So a couple of years ago, Arnadottir Chalfie and his colleague put forward 4 criteria for a channel to qualify as a mechanotransduction channel. Now, as you know, Marty got a Nobel Prize for his work on GFP, but his career's work is about mechanosensory channels. So the 4 criteria are: first, the channel must have appropriate temporal and spatial expression patterns within a mechanosensory organ. Second, the channel must be required for the animal's response to the relevant mechanical stimuli. And alteration of the properties of the channel protein should alter the properties of the mechanical response. And lastly, the heterologous expression of the channel should demonstrate that it could be gated mechanically. So over the years, quite a few candidates have been found for as mechanosensory channels. And as illustrated in this very recent review by Marion Goodman and colleagues, there are several prime candidates that are very good case candidates. One is the amyloid sensitive sodium channel. There's some of the trip channels, the PMC, and the recently discovered Dscam. Now, remarkably, as of 2010, when the review was written, not a single eukaryotic channelopathy involving mechanosensation has been reported to satisfy all four criteria. But this changed this year when Adam had the potions that they showed quite convincingly that Dscam has satisfied all four criteria. But nevertheless, it's very, very rare to satisfy all four criteria. So I'll return to that in a few minutes. But before doing that, let me introduce you to the experimental system we've been using and explain how we got into this mechanosensory field. So for the past 12 years or so, one major focus in our lab has been the study of dendrite morphogenesis by using a group of Drosophila larval sensory neurons known as Dendritic arborization or DA neurons. And those neurons are organized in a bilateral symmetric and segmentally repeated pattern. This is in Drosophila larva. And based on dendrite morphology, those neurons can be divided into 4 different classes from 1 to 4. This is from simple to complex. Okay. And by using those neurons, my colleagues and I have gained some insights about the control of dendrite development. And we have learned, for instance, how axons and dendrites are made differently, and how a neuron acquired its neuronal type-specific dendritic fiber, how dendrites of different neurons are organized relative to one another, how the size of the dendritic fiber is controlled, and how the pruning and remodeling of the dendritic fiber is regulated. So if we take the class 4 DA neuron as an example, so we made a reporter which is specific for class 4 DA neuron, and that allows us to visualize the dendrite in developing living larva. And you can see it's really quite a pretty pattern. And the dendrite of those neurons cover the entire body wall in a very regular array. So how does this beautiful array of sensory fibers develop? And we know now that several interactions are important in shaping the organization of those dendritic fibers. I'll just mention a couple very briefly. One is that there is repulsion between sister dendrites and a phenomenon known as self-avoidance. And this is mediated by a cell recognition molecule known as Dscam. So the work on Dscam from our lab was done by two postdocs, Peter and Seagram. And there was parallel and complementary work from the labs of Grueber, Tversky, and Schmucker. So basically, if you look at any DA neuron, or for that matter, any neuron implied, the dendrites tend to spread out. They don't bundle together. And this is due to sister dendrites repelling one another via the Dscam. So in the Dscam mutant, what you see is the dendrite will bundle together or cross over due to defects in self-avoidance. And that applies to class 4 neuron as well. And another type of interaction is known as timing. And so the class 3 and class 4 DA neurons, they exhibit timing. Let's take class 4 as an example. There are 3 class 4 DA neurons, and the dendritic fiber of each one occupies about 1/3 of the hemisegment. There's very little overlap between dendrites or adjacent neurons, class 4 neurons. And this arrangement is known as tiling because the tiles covering the floor. And this was initially discovered in mammalian retina, presumably to ensure complete and non-redundant coverage. And timing is caused by homotypic repulsion between adjacent dendrite or adjacent neurons at the same time, as indicated by this ablation experiment, in which we ablate one of the classical neurons shortly after it's born, and you see that the neighboring classical center dendrites will invade the vacated area, as you would predict from a like mechanism. So by using self-avoidance, timing, and several other mechanisms I won't get into, so the larva was able to make this regular array of sensory dendrites that cover the body wall, have the body wall. So then the question is why? Why did they go through trouble to make such a nice array? And for that, then we need to know the function of those neurons. And based on work from several labs, from Benzers, JC, Therapeutics, and our lab, we now know the classical DN neuron, they function as polymodal nociception neurons, and they can sense high temperature, harsh mechanical stimuli, noxious chemical like wasabi, and harmful light. So the last one was found in our lab, and this work was published over there. I'll just give you sort of the summary. So basically, to our surprise, that those DN neurons are actually photoreceptors. And people have been studying blood for over 100 years, and there are still surprises. And those new, so before that, it's known that Drosophila has the so-called larval eye. They are called Bolwig organs. Those are photoreceptors in the head of the larva, and its function is to sense low light for avoidance, and they use rhodopsins as a photosensor. And this newly found photoreceptors, they cover the entire body. But their job is to sense strong harmful light, like shortwave, intense blue or UV light, and it uses an entirely different sensory transduction system. And so this may explain why those dendrites have arrangements so similar to how nerve fiber in the retina, because this will allow the larva to detect harmful light in any part of its body, so it can get out of the way. And it is this same classical DN neuron, that's where Therapeutics demonstrated that that the piezo is a channel that mediates harsh mechanical stimuli. As they reported in their paper earlier this year, that they could satisfy all 4 criteria that I listed early in the talk for it to be a mechano-transducing channel. Now, they also found that those classical DN neurons, while they can sense noxious mechanical stimulation, they do not sense gentle touch. Okay? So for the rest, in fact, very little is known, for instance, in mammal, what the molecular mechanism underlying touch. So for the rest of my talk, what I'm going to do is to try to address this question. So which neurons and what kind of mechanotransduction channels are required for sensing gentle touch in the software? Okay. So, as I already mentioned, classical DN neurons, their function is nociception. And what we found is class 3 neurons are the ones that are responsible for gentle touch. And let me give you the evidence. So for gentle touch assay, we use the one that was developed in Zucker's many years ago. So you simply take a little hair like from an eyelash and touch the side, anterior side of the larva gently, and then score the strength of the response, ranging from 0 to 4. So 0 being there's no response, and 4 being the most extreme response, which is multiple waves of a reverse locomotion, and it could be anything between. And so for each larva, we test 4 times and add up the score. So next, to try to determine which types of neurons are responsible for this gentle touch response, so what we did is imply this so-called GAL4 UAS system. We can use neuronal type-specific GAL4 to drive the expression of mechanotoxin to block the synaptic transmission. So what we found is if we drive the expression of this toxin in class 3 neurons to block synaptic transmission, and then there is a significant reduction of the response to the gentle touch. But none of the other classes of DA neurons have this property. And so next we wanted to know whether the class 3 DA neurons, they can actually respond to gentle touch. So we did 2 kinds of experiments. One is that we use the Dscam, which is sort of a new and improved calcium indicator developed by our collaborator Grueber at Union Park. So we put that in those neurons, and what we found is if we touch a body wall gently, then the neuron that innervate that part, the class 3 neuron, will show a strong calcium response. And to better characterize the physiological response, we did focal extracellular reporting from the soma of the class 3 DA neuron. They use another pipette to gently touch the body wall. And what we found is that even a very small stimulus, which is only cost like 10 microns, indentation of the body wall is sufficient to elicit a burst of action potential. And the number of the action potential increases with stronger stimulation. And if we give a sustained mechanical stimulation, the neuron adapt very quickly, and then there's an off response. So based on all those experiments together, we can conclude that class 3 DA neurons, they are rapidly adapting low threshold mechanoreceptor mediated gentle touch sensing in right logic. So now we know which cells are responsible. The next question is, what is this the mechanotransduction channel used in this case? And the answer is NOM-C. And what is NOM-C? So NOM-C again was identified in Zucker's lab quite a number of years ago from their screen for uncoordinated mutation. And they also found that the mutant adults have reduced bristle mechanoreceptor potential. So NOM-C is a founding member of the TRPN family of tryptanyl. And what's unique about this family, it has a very long stretch of anchoring repeat. And NOM-C is also known to be important for hearing in fly, adult fly, and visual is also important for proper locomotion. So to study this protein or this gene, Chen-Li in the lab, she generated a number of reagents. In particular, she isolated cDNA, which is different and longer than the reported sequence. And the important thing is that the cDNA she isolated, we can show that it's functional because it can rescue the mutant phenotype. And with the antibody against NOM-C, well, we found that indeed that the class 3 neuron express NOM-C at high level and not the other DA neurons. And it has quite interesting cellular localization, which I'll come to at the end of my talk. So next, then we look at the NOM-C mutants touch sensitivity. And what we found is that in NOM-C mutant, their response to gentle touch is significantly reduced. And importantly, this phenotype can be rescued if we just put the non-C, the full-length non-C clone, back only into class 3 neuron. This we can do by the appropriate code drivers. And so what this experiment told us is that non-C is required self-autonomously in class 3 DA neurons to mediate genital touch. So next we ask, what is the physiological response of the mutant class 8 DA neuron, the non-C mutant class 8 DA neuron? And if you look at the morphology of those neurons, they are perfectly normal. So it does not cause obvious developmental or morphological change. However, the non-C mutant, if you look at the class 8 neuron, they don't respond to genital touch anymore. And so here's the stimulation, and there's no burst or action potential. But again, if we put non-C back only in class 3 neurons, we can rescue this phenotype. Now the neuron regain its ability to sense genital touch. And this is sort of the dose response curve. So this is wild type with increasing strength of stimulation. There are more action potential firing. But in the mutant, it's essentially insensitive. But put the non-C back into the cell, the class 3 neuron is restored. And likewise, the calcium response is also disappeared in the mutant, but can be recovered if we put non-C back into the class 3 neuron. All right. So far, we have satisfied the first two criteria. That is, non-C is expressed in the right cell in class 3 DA neuron, and it is required for class 3 DA neuron to respond properly to genital mechanical stimulus. So the next question is, now, if we ectopically express non-C in a neuron which normally is not touch sensitive, can we confer the ability to sense genital touch in those neurons? And for this experiment, we use class 4 neurons. Report that they are nociceptive, but they do not respond to genital touch. In fact, in parallel experiment, if you look at class 4 versus class 3, with increasing stimulation, the class 3, but not class 4, will fire action potential. Right? And normally, class 4 neurons, they do not express non-C. So this is green labeled class 4 neuron, and this is non-C staining. The staining you saw, those are the dendrites of neighboring class 3 neurons. And if you look at the merged picture, there's no overlap of the staining. But if we use a class 4 specific driver to express a non-C in class 4 DA neuron, now those neurons express high level of non-C in its soma and its dendrite. Those neurons now gain the ability to respond to genital touch. This is a reporting of action potential, and this is the calcium response. In both cases, now the neuron can respond to genital touch. So from this, we can conclude that non-C ectopic expression in normally touch-insensitive class 4 DA neuron is sufficient to endow them the ability to respond to genital touch. So next we want to know, is non-C a channel? And in particular, is it a core-lining subunit for mechanical sensitive channel? And this has been a stumbling block for many of the studies of the mechanosensitive channels, because often it's, for whatever reason, it's hard to do that. But fortunately, in this case, we were able to do it. And so we found that if we express non-C in cultured Drosophila S2 cells, which normally does not express non-C, and then we can see non-C induced channel activity in whole cell patch. So this is a non-transfected control or cell transfected only with GFP. there's no obvious channel activity. However, with non-TGFP, now you see those spontaneous channel activity. And those are non-selective cation channels, which is permeable to sodium, potassium, cesium, and calcium. Now then, can those channels be activated mechanically? And so for that, we use two different methods. One is that we use a polished glass pipette and link to some piezo device. We can apply defined mechanical stimulation to the S2 cell, and while it's being recorded. And indeed, you can see strong mechanical activating channel activity, and there's also an off response. And this, the strength of the response depends on goes with the strength of stimulation, and also with stronger stimulation, the latency drops to, and the latency is less than 2 milliseconds, so it's too short for second messenger channel mechanism. So consistent with it in a directly activated channel. Now, an important question is whether the spontaneously active channel and the mechanical gated channel are they the same? And we think they are. So because I'm sorry. So this is the second method where we apply negative pressure to a petrol membrane, which is under outside our pack before decondition. We saw a high-speed pressure fan, and again we could see stimulus-dependent response. So the higher the negative pressure, the stronger the response. So I was saying that important question is whether the spontaneously active and the stretch activated non-C channel are they the same. We think they are the same because they have the same unitary conductance, and both they have very similar pharmacological properties. So both are blocked by one of the chip channel blockers, and both are blocked by FM143, which is a very effective blocker of the hearing channel. And so based on those, so I should say those blockers are not terribly specific, but the important point here is that the spontaneous active channel and the mechanical activated channel, they have very similar properties. So the experiment I've described to you so far says that the non-C, when transfecting the cell, can induce mechanical gated channel activity. But that doesn't say itself is a purulanin subunit, because it could be recruiting some other subunit from the S2 cell. That's actually the purulanin subunit. So to ask this question, ideally one would like to purify the protein, put it in lipid bilayer, and show that it can be mechanical gated. We haven't tried that experiment. So we did the next best thing, which is to make a single amino acid mutation in the predicted purulanin region and show that it changes the property of the channel. So the non-C, just like bigger channel, you have predicted purulanin region between S5 and S6, and we found that a mutation at this position would significantly reduce the single channel conductance as well as change the IV curve. And so those experiments support that this is itself as a purulanin subunit. And again, we asked whether the spontaneous active one and the mechanical and stimulation activated one are whether they have similar property, and they behave very similarly. In terms of the change of the channel property in single channel conductance as well as IV curve and the activity. So we think a possible explanation here is that even though the S2 cell sitting at rest without any added mechanical stimulation,